Okay, and here's an example of why that sort of thing can be useful. We've got two sister species, Drosera stenopetala and Drosera uniflora. Their present distribution is, the stenopetala is New Zealand. Present distribution for uniflora is the southern tip of South America, Patagonia, and a bit up into Chile. So, these are sister species, and these are well sampled. We know that these are sister species based on strong genetic evidence. So, a simple question might be, where's the ancestral area for these two species? Because we know that they had a common ancestor. We've got a dated phylogeny. We know that their ancestral, their most recent common ancestor was from about 8 million years ago.
We have fossil evidence?
No, not for those species. But we can say, we know the environmental preference for this. We know the environmental preference for this. We can reconstruct our ancestral environmental preference, and perhaps that will give us some information about where was environmentally suitable for our putative ancestor at a relevant time period. So, we develop our model and say the temperature preference for this species is this. Obviously, we're going to use more than just temperature, and the temperature preference for this species is this. Okay? Temperature is this. The age for the most recent common ancestor is somewhere between 8.5 and 8 million years. We can then take that model and we can project it into our reconstruction for 8 million years ago. And we can say, where's environmentally suitable? Well, some areas of Greenland and New Zealand. Okay, well, what do we think is plausible in terms of an ancestral area based on our climatic suitability? Greenland, Alaska, or New Zealand?
Denmark.
Possibly. Yeah, but so this is a line of evidence that suggests that using ancestral state reconstruction, we can work we can calculate environmental niche parameters for our ancestor, and we can say that using those set of environmental preferences, it looks like New Zealand was more suitable than any other area.
And here you used median or something instead of the extremes?
No. So I just presented that as a simple example, but we used the minimum and the maximum temperatures. We used the precipitation preferences. We used a subset of bioclimatic variables. Yeah.
How was when I used something like the Phytools package? We know that
Yeah.
to do like evaluation of climatic variables, I make a PCA. I think I will have to do for every like PCA. I have to do 1, 2, 3. I have to do a separate run. I cannot like you say I do both temperature and precipitation. How do you do that in one?
No, so you have one analysis that is ancestral state reconstruction for minimum temperature, another one for maximum temperature, another one for precipitation in the
You do not combine them.
Okay, so once you do that, you have values for each of the nodes, then you can put those numbers together, and that creates your bioclim model that says that temperature minimum maximum is this, precipitation minimum maximum is this. And then you say, okay, well, that given those parameters, where is environmentally suitable at a specific time frame?
Okay. Yeah.
Right. So that's the theory, and here are some examples of this sort of principle in action. So Smith and Donahue, 2010, they did some another thing with. A couple of years earlier, but they reconstructed the ancestral niche for these species, projected them into paleoclimate reconstructions for different time periods, and used it to estimate potential ancestral areas to answer questions about biogeography.
And so here presumably they did that for not extreme values, but like the best value and tolerance to departure from extreme, or?
Yeah, so in this case, they didn't use a biotin model. What they did was they developed profiles for, they developed Maxent models, and then they used the Maxent models to develop response profiles
Yeah.
for each variable.
Yes.
And then they used a different ancestral state reconstruction technique that reconstructs profiles
Oh, really?
across
Across the
the
variable?
trees. Yeah, yeah. So instead of just the minimum and the maximum, essentially on their tree, they had like this
Uh-huh.
and like this, and then they reconstructed this.
Got it.
Yeah. I don't agree with the maths that does this, but it's a different and more complicated methodology. But it essentially does something very, very similar. You start with some values associated with each species. You reconstruct, you project into ancestral. So you project your model for your ancestral lineage into paleoclimate reconstructions for the appropriate time period to say where, at different points in the past, appears to be environmentally suitable. And I'm going to give one more example, and then I'm going to talk about all of the problems with this kind of modelling. The ancestral area question is it's a very common question raised in biogeography, and the methodology that has been used previously to try and answer this question has been entirely based on geography. And so this is something that was in Town's talk earlier, that the traditional method of investigating biogeography is based on present-day distributions and trying to work out ancestral areas based on what we can see from present-day distributions. And so the traditional way of doing this is again using a different kind of ancestral reconstruction, but ancestral area reconstruction, where what you do is you take your present distributions. This is for cyclamen, a small flowering plant that's found around Australia. Okay? And you group your distribution into areas. So this is zone F, which covers all this area, and here zone I and zone B, based on sort of continuous areas of distribution. Okay? You map those onto your phylogeny. So C. elegans is in area F. Okay? And then you can use ancestral reconstruction for these characters to say that in the ancestral area, this that's F and this that's F is also F. Okay? Whereas the ancestral area for this node is F and G, because that's where these things are found. Okay? So this methodology is called DIVA. Dispersal something vicariance analysis. I forgot what the I stands for. There's a slightly better version of this, a more updated version that tries to incorporate dispersal attention between areas and putting barriers in, called Lagrange. But essentially what they do is they reconstruct ancestral areas based on what we observe in the present day, and if we've got a nice pattern, we can end up with a single selection for our ancestral area. So for all of this clade, we reconstruct area F as our ancestral area, even though it's found in area F and G and J. Okay? But what we can't do is we can't suggest that the ancestral area is here, or here, or here. Okay? So we're restricted to only considering areas where we currently see the distribution. So that's, so we can get some interesting answers from this, but it's not necessarily realistic because the assumption is that ancestral areas are a subset of where we currently see our organisms.
And it's not correct. Some species.
No, so it has difficulties, yeah? The assumption that things haven't moved is quite a difficult one to justify, but if we're just looking purely geographically, then this is the best that people have come up with so far on using this sort of methodology.
And even with Lagrange, which is allowing dispersal events to previously unoccupied areas, it's still thinking about the world as if the world has been basically static,
Yeah.
right?
Okay. So, and what you end up with these sort of techniques is you often get this sort of clumping of areas at the base that basically says the ancestor was everywhere and then population was divided. Okay. So, this is biogeography based on current geography, and there's lots of good things about this, and so I don't want to just sort of say that okay, this is no good and then ancestral niche reconstruction is in some way better. It's different because we're making inferences about ancestral areas based on environmental requirements and not based on just geography. Okay, so here's the same group. Okay, it's the same phylogeny just presented differently, and we can use our BITC models to look at present-day environmental preferences. These are sort of amalgamated together for this clade, and then we can look at our ancestral reconstructions, and then we get back to our ancestral cyclamen, and we can project that into a relevant time frame, which also happens to be 10 million years ago. It's quite lucky that I found examples that could fit into the paleoclimate reconstructions that I could get hold of, which was 8 million and 10 million years ago. And this is the sort of area that we reconstruct. So this is, you might not be able to tell, but this is Europe. This is the UK. This is France. This is Spain. Okay, what do we sort of look at that and we see? It looks quite different to modern day geography. Partly, it's because the very coarse resolution of the underlying data, and secondly, it's because geography was different 8 million, 10 million years ago. Okay? But what we can say is that the grey areas are the sort of grayscale, black, dark grey, light grey, are our BITC model, where the black represents all of the environmental parameters that are within the range of our reconstructed niche. The light grey, one of them is outside, but most of them are, and the really light grey, maybe just one is inside. I can't remember the numbers. I think we've got a 5 and a 4 and a 3 there. Okay? And then the green areas are completely outside of our reconstructed ancestral area. Okay? So it's really fragmented, but it's a different interpretation of ancestral area based on our niche reconstruction. Okay, so there are many, many, many issues that we have with this kind of modelling. And because we're combining a whole bunch of different methodologies, each of these methodologies comes with its own set of caveats and things that we need to keep in mind when we're using them. Phylogenetic reconstruction, okay, is an estimate of relationships. It's not the real thing. And our phylogenies change as we learn more about the species, as we get more sequence data, as we get better sampling, as we find new species that weren't on our phylogeny. Okay? Obviously, if our phylogeny changes, our ancestral state reconstructions are going to change, because nodes that are in one phylogeny are not in another phylogeny. Yeah? And the structure of the phylogeny changes how our reconstructions work. Okay? Some people completely disagree with reconstructing environmental characteristics of species on phylogenies. Also, the same people tends to not like dating of phylogenies. Okay, so when we date phylogenies, as Tom sort of mentioned earlier, we draw our phylogeny and we set an age on it, and we look at that and we say that node is 8.52 million years old. But actually what we're saying is that's 8.5 million years old plus or minus 2 million years. Okay? So really what we've got is something like this in terms of our age range uncertainty. And we saw that on the Smith and Donahue phylogeny. Okay? So we can put an age on our ancestral nodes, but that comes with an error. And you might remember that our paleoclimate reconstructions come with an error as well. Okay? So we've got an error here, and we've got an error associated with our environmental data, and they might overlap, and they may not. Okay? And, we've been emphasising how much debate and criticism there are of different modelling methodologies. Well, the same thing is true of molecular dating of phylogenies. Okay? There's just as much really heavy criticism as there is in the field of niche modelling, only it tends to be more vociferous and personal. Dating our phylogenies requires calibration, and that calibration requires, generally speaking, fossil evidence. We need to find a fossil, stick it on the right part of our phylogeny. So we need to be able to identify this fossil as being the ancestor of a bunch of other things. That's not easy, because most fossils are incomplete. We don't have a complete structure, and it's difficult to position it accurately on our phylogeny. Okay, and as I've already said, ages assigned to nodes have large error margins. So, our phylogenies come with a whole bunch of uncertainties. Our paleoclimate reconstructions come with a whole load of uncertainties. We have different models reconstructing different environments. Okay? The data is low resolution. We have this issue of non-analogous climates being evident in the past, and we don't see it in the present. And we have a large error margin in terms of the age that we can assign to our paleoclimate reconstructions. Okay, so our niche model. All of the standard criticisms of niche modelling applies here. We have to develop a present-day model. But because of the restrictions that we have, we're using a relatively simple model. Okay, people tend not to use bioclim models, and we've talked about the reasons why not over the past 2 days. But if we are reconstructing one variable at a time on our phylogeny, we have to have the assumption that each of these variables are independent, and that's the bioclim has that assumption.
But using that approach that you mentioned there, that you draw there in that whiteboard.
Yeah.
Yeah.
Wasn't it possible to apply it to a surface instead of being a line, multi-dimensional surface?
No.
It's not possible?
Well, people haven't done it yet. But even there, this curve is our response to temperature. Okay? It's not, we have another phylogeny that has another curve that's a response to precipitation. So it's still treating each variable is independent of each other. Okay?
Yeah, but then those could, if you had made a accent model that didn't include product features, you could, and then you reconstructed your values to the individual variable, your curves to the individual variables.
Yeah.
You know, those could be applied, I think, into the original accent model.
Yeah.
I don't know.
I guess they could, but nobody's done it.
Not even the Smith and Dunn.
No. What they did is they reconstructed this profile, and then they used this profile and the next profile in a bioclim type way
Oh.
to so they didn't use that to feed into a new Maxent model. Okay. Models are simplistic in comparison to those available for present-day modeling. Yeah. Models treat environmental parameters as independent. We reconstruct each environmental variable separately on a different biology. And there's an extremely long time period between observed data and inferred distribution models. Okay, so we've got all the issues of transfer from one time frame to another. Okay. Let's think for a moment, and something I haven't written down, about variable selection. How do we select our variables for our models? We spend time thinking about what variables are suitable for this species, yeah? And then we choose a subset of all the variables we can potentially have and build our model. But if we are reconstructing environmental characteristics across a phylogeny, each one of these species must have its present-day model built with the same environmental variables. That's a restriction that we have on this kind of modelling that we don't have for individual species models. Okay? Because our ancestral model has to contain the environmental variables that we can see for all of our present-day species. Okay? So this is an extra constraint that all of the species in our phylogeny, we have to use the same environmental parameters to start with. Yeah? So that violates one of the golden rules that we had for our species modelling, that we need to tune for each species the right set of environmental variables. Okay, so there's a very, very long list of caveats that we have, and sometimes I sort of feel that we're sort of building model upon model upon model that is really just a house of cards, and one little card falls and everything falls down. Yeah. So only one of these many, many issues needs to bite us for the whole thing to just have freedoms. Okay. So we've got a difficult. It's a difficult problem, and I don't in any way suggest that this approach is an optimal solution, and people are improving it all the time. But it's an approach that gives us an answer, and if we have fossil distribution data, we can potentially validate our models by saying, we've got fossils for this ancestor, we found it in this period, in this location. Does our ancestral reconstruction model select those areas as being suitable?
Sorry, Chris.
Yeah.
So you have to check, how do you solve the problem of the variables?
Well, you can't. So, how do we select variables for this? Well, we know that we are restricted to saying that we have to have the same set of variables for all of our species. Okay. What's the underlying assumption of our ancestral state reconstruction? What do we say? What do we assume about our niches on our phylogeny? We assume that there's some phylogenetic conservancy. Yeah? Okay. But for any variable, we've got tests that tell us whether that variable appears to show phylogenetic conservancy. Yeah? Well, surely we can test each of these variables in turn, and we can use those variables that show phylogenetic conservancy, and we can discard those that don't. Because an ancestral state reconstruction with variables that don't appear to show phylogenetic conservancy doesn't seem to be a very good idea. Okay? So that's one way of restricting the variables that we use. Okay, so that we're sure that the variables that we are using are showing phylogenetic conservancy, so an ancestral reconstruction with those variables seems to be a sensible thing to do. Or at least not a stupid thing to do. Okay? So that's one way of choosing variables. But that chooses variables that fits our assumption of ancestral phylogenetic conservancy. It's optimized for that. It's not optimized for modeling. It's not optimized for niche modeling. Okay? So we've got these conflicting reasons to choose variables. One is we should use variables that show phylogenetic conservancy because then our ancestral reconstruction makes sense. But on the other hand, we want to choose variables that adequately describe the distribution based on our present day using the sort of methodologies we use for choosing variables for normal niche modelling. So we've got these conflicting things, and if we're lucky, or we do a lot of work and we look at a lot of variables, we find some that sit in a nice overlap between those two. But that's quite hard to find. So this sort of thing is quite hard work. It takes a lot of time and effort, and it's not a nice software package that does it all for you. So it's a big task with lots